Second, for those who joined us today, last week was an opening lecture, so it was softer and more easy. So if you feel that today's talk is complicated, you have to go back to the website. And Mo will explain everything about the website.
Okay, so the website is the Moodle website. I'm sure you all know about it. If you cannot register or something, just come to me after the lecture. I will sign you in. All the presentations will be there a few days after the presentation. And we will have some articles and papers, relevant ones, for you to read. If you need something to read before, if you're not from biology exactly like you guys, and if you need some other guidance, come to me and I'll give you other papers. But any problem you have, write me an email. My email is there in the website.
Okay. Okay. So we are getting back to the biological basis of Alzheimer's disease and the genes and interactions that are relevant. And last week we talked about the basic impairments in sporadic Alzheimer's disease. And now we want to focus on the genes and pathways that are involved, the animal model mice that are available for studying this disease, and the current and future therapies. What is done today, what is explored as preparation for the future. Okay. And let's start with the last topic. So let's talk about therapeutics. Yeah. כמובן, לא שם. פה זה רק המצב. שם זה פשוט שכחתי. Okay. So we talked last week about the different aspects that are known to be impaired, and as is often the case, based on the impairment, pharmaceutical companies and pharmacology researchers are trying to develop drugs that will compensate for the loss or correct the impairment. So kinases, we talked about kinases last week, enzymes that induce phosphorylation of other proteins. Kinase activities are notably impaired in Alzheimer's disease. And you see here that kinase inhibitors are tried, but I would like to add a subtext to them. Kinase signaling is badly impaired in different tumors. There's a lot of kinase inhibitors that are already used and quite successfully in blocking the development of different cancers. And the companies keep looking for other indications. And I believe that this is the reason for the effort to develop kinase inhibitors also for Alzheimer's disease. So far, I don't know about even one of these drugs that was really effective. The next line talks about tau fibril formation. Now remember, for those who were not here last week, we talked about the tubulin-associated protein tau that is hyperphosphorylated. And here you need a kinase inhibitor if that would be the cause. And you want to prevent the formation of the fibrils due to tau hyperphosphorylation and dissolve the pre-aggregates of such fibrils, which is really very difficult today. Again, one of the major topics that we covered last week was that the main problem with neurodegenerative disease, Alzheimer's leading, is that we confront diagnosis far too late. To reverse disease problems is much more difficult than to prevent the acceleration of such events. So people are trying different, very creative agents. One of them was developed in Tel Aviv University. There is a startup company trying to change the fibril formation by tau. And again, so far, none of these drugs has been approved for human use. The next topic is interesting, and we didn't talk about it much last week, chaperone systems. Now, a chaperone, as we know, is the lady who is accompanying a young woman to avoid creation of unwanted contacts. The chaperones in biochemistry are proteins that accompany other proteins, preventing them from forming an unwanted contact. And in more accepted terms, the chaperone system is involved in controlling the folding of proteins, in preventing the improper folding of proteins, which is the reason for the creation of these unusual contacts, and mutations in these systems are known in all diseases, not only neurodegenerative diseases. So again, you see here that development of drugs is driven not only by the biological basis of the disease, but also dependent on the availability of drugs based on know-how in other fields. So people are trying to prevent the chaperone. Actually, there was a seminar today. Pierre Golubinov was a faculty member here, and then he moved to Lausanne in Switzerland, and he's an expert in chaperone mechanisms, but mainly in yeast, not in Alzheimer's. Okay. Then stabilization of the microtubules. This really is addressing the very biological basis of Alzheimer's disease. If formation of neuronal fibrils, of tangles, if abnormalities in microtubules, which are the cytoskeleton, the skeleton of the nerve cell, if that is a problem, then let's try to stabilize the correct structure. And that could or should prevent the deterioration in the disease. So again, it relates to tau, and it sustains axonal transport because the neuronal cytoskeleton is also important for maintaining the capacity of the nerve cell to produce proteins in the cell body and send them all the way down to the synapse, which may be very further away in cellular terms. And amyloid-directed therapies. If the fibril formation by amyloid and neurotoxic amyloid peptide is the problem, then what we need to do is prevent the amyloid peptide from creating the fibrils and from contributing to the tau abnormalities. Next, attenuating inflammation. We mentioned it last week, we talk about it today furthermore. And the involvement of inflammation is today realized as an important contributor in many different diseases. Again, not only neurodegenerative diseases. And attenuating inflammation is considered as a potentially helpful approach. So what you see here is all tau-related medications. None of these is still functioning. The major medications that are available today are targeted at the cholinergic basis of the disease, which was the first to be developed. And here again, we need to talk a little bit about the dynamics of therapeutic studies. Yeah.
I may ask two small questions,
Sure.
but not really from this field.
Sure.
But there was work by Andrew Dillon and Ehud Cohen from our university that showed that IGF signaling can attenuate Alzheimer's very nicely in both nematode and in mice. Would you classify this under any of these?
The IGF signaling, the closest is for inflammation.
And the
But
second,
not to tau.
there was a recent paper in Science where they used, again, an anti-cancer drug, as you mentioned, that targeted a signaling pathway downstream of it, and they showed very nice results in mice.
Yeah. Okay.
Right.
So this is exactly what I was getting to. We need to look at such tables in the context of the time scale that it takes to develop a therapeutic. The Alzheimer's disease had been recognized as a cholinergic deficit in the early 80s, and then companies started developing drugs. And it takes about 15 years from the beginning to end of a successful drug being developed until you start using it. And sometimes you start using it and within 5 years patients show complications and then it has to be withdrawn. So we are talking about a scale of 20 years. All of these drugs against tau were started to be developed in the 90s because that was a later discovery, which means that we can't really expect yet these drugs to be available. What L did was 3 years ago. No question about that. It will take 15 years more.
But previous work trying, not yet successful, but trying to target this pathway, and then you can say, well, let's use this drug to target.
Exactly. So that cuts it down to 10 years, not.
And they took already in the science paper a drug which is already approved. So you can skip already to phase 2. You don't need to do preclinical, you don't need to do phase 1.
Well, you need more preclinical studies for the FDA to approve the testing of an already approved drug for one indication for use in another indication. Okay, so you cut down the time about 5 years, but not more than that. So all of this needs to be taken in that context. There's another problem. I was once lecturing in Roche in Switzerland, and my host said, what do you think is the investment in the preclinical research out of the total cost of drug development? Anyone with a guess?
Nothing.
Percent.
What is
5.
this?
2%. So everything that is done in the lab, and we think it's very expensive, and indeed it is in our scale, is about 2% of the cost of developing a drug until it gets to the market. Most of the expense goes to people who advertise it, to public relations, to negotiations between experts and the FDA, things that we are really not experienced in. So this always keeps amazing us, but that also implies another thing. When a company develops a drug, it doesn't want to know about new developments. To the extent that companies sometimes try to block new developments. There are a lot of stories about companies signing a contract with a university on a totally new invention, and then they do what is called professionally shelving it. They put it on the shelf to avoid any competition between that new invention and their own drug. There are lawsuits in Israel today against Teva exactly on that account. So everything we see here, it looks like pure science. There is nothing like pure science, I'm afraid. Anyway, the next thing we talked about was the ApoE. We talked about the apolipoprotein E being a gene which modulates the disease process. So it is not causing it, but it confers protection, and that protection may be more or less efficient depending on the gene variants. So there are 4 major variants of the ApoE gene, and variant number 4 is bad news. Carriers of 2 copies of variant number 4 are at considerably higher risk to come down with Alzheimer's disease, and that created a whole new issue of an ethical problem. Nobody can change that composition, and a lot of genetic studies offered people to let them know whether or not they carry this risk, except that they can't do anything about it, which is an ethical Again, the ApoE pathway is relevant for different mechanisms, and here what you see is the connection between the apolipoprotein E mechanism and amyloid beta. But if you talk to someone interested in tau, they will tell you that it is highly relevant for the creation of the toxic neurofibrillar tangles. And if you talk to someone interested in cholinergic mechanisms, they will explain that cholinergic neurons are more susceptible than other neurons to problems of related to ApoE functioning. So what the message I'm trying to convey is that there are facts and different beliefs and trends and traditions, and all of the mechanisms are probably correct, and all of them together are interlinked and contribute to the disease. Okay, so why ApoE? The entire family of the LDL receptors is very well conserved in different species. And what you see here are examples in Drosophila, in flatworms, in Caenorhabditis elegans, and in mammals. So this, just by this evolutionary conservation, this emerges as an important mechanism of interaction between cells and a safeguard of the well-being of cells. Which are the mechanisms associated with Alzheimer's disease that could be affected by changes in the apolipoprotein E mechanisms? And this review summarizes it beautifully, and they practically mention everything that we know that could be relevant. So cell migration is, of course, important. Axon guidance is a mechanism that refers to the way that a nerve cell sends its processes to connect to another cell. And correction of impaired connections is, of course, an important mechanism when one loses nerve cells. And the bad news is, of course, that we all lose between 10 to 20,000 nerve cells per day from the age 20 on. So axon guidance is an important process not only in neurodevelopment, but also in the adult life. The microtubule stability we discussed, so this is relevant for the tau mechanism. Synaptic plasticity would be the mechanism referring to the capacity of an interconnection between nerve cells to change its efficiency of functioning. There's a Nobel laureate that is a friend of our institute, Eric Kandel, and he's a great lecturer, and he gives a seminar, and at the end of that hour he says, if you listen to me carefully, the structure of your synapses has changed. So that is synapse plasticity. Changes, facilitation, enhancement of the capacity of the synapse to send messages, which of course would contribute to learning and memory. Then we have regeneration. We just talked about the need for neuronal regeneration. What you see here is that regeneration also involves not only the axon, which is this, but also the myelin wrapping around nerve cells, which is the insulation. So you need to send signals, and you need to protect the wires that include and submit these signals from the environment. Anyone here who knows which disease is accompanied by damage to this insulation? MS. Bravo. MS. Amyloid deposition, we talked about, and finally the survival of neurons. So all in all, they refer to everything but the cholinergic mechanism, which was known beforehand and was considered by then as less important, not news really. And then they tried to break down the effect on amyloid metabolism of as those are conferred by the ApoE. What we think about is the wrapping through lipids of compounds that could serve for gain of function and for loss of function. It could serve for sending materials from one side to the next, or for wrapping those toxic materials that we don't want to be in touch with the nerve cell. And we are talking here about three basic types of cells in the brain. We are talking about the nerve cells, which have the LDL receptors. We're talking about the microglia, which are the representatives of the immune system in the brain. And they could be coming from two sources. Microglia in the brain could be cells that were born in the brain, developed there, and serve their immune function, and that's what they normally do. Or they could be the cells that were before macrophages, that got into the brain through the blood-brain barrier, which happens more often under stressful insults, for example, and then they acquire within the brain the properties of microglia, which again keep their immune functions, but in a different flavor from what they did before. And astrocytes would be the cells that are there. We used to call them glia. Now the glia are, of course, classified to different subfamilies. The astrocytes are the major family of those. And what they can do is wrap around a precipitate of toxic amyloid and just put it away out of the system and avoid the damages that it does. In addition, the astroglia have receptors. For example, they have acetylcholine receptors. They have glutamate receptors. They can react to signals in the vicinity of the nerve cells. So nerve cells are special, but not the only ones that can react to signals. And precipitation of plaques, these are the plaques here, can happen not only next to nerve cells, but also around the blood vessels. That is, for example, the disease that was referred to when Arik Sharon's condition was discussed on the radio. They said it was not noticed that he suffered from a vascular disease. Multi-infarct dementia is a disease that is from the Alzheimer's family, but it's not the classical Alzheimer. In that disease, the plaques develop mainly around microvessels, and they create a lot of foci for little strokes within the brain. When there is a hemorrhagic stroke, as was the case with Sharon, this becomes really devastating. What about inflammation? We see, yeah.
Sorry, I'm a bit lost about
Okay.
how ApoE was related to the last slide. Like what?
Okay, so you need to read the basics on the apolipoprotein E function. We need to look for a review on that. And Danny Michelson will talk about that in a whole lesson. But it's a good idea to read. The lipid metabolism is a basic constituent of biology, and there's a lot of material in any textbook on ApoE mechanism of action. Okay? Fine. So now we're talking inflammation. We can talk about inflammation as damage. And this is the context in which it is mentioned with regards to Alzheimer's disease. But we can also talk about inflammation as a stress reaction of the brain in terms of getting ready for damage. it's confronted with any stressful insult is to get ready for injury, because we evolved from organisms where a stressful condition was a threat for survival. And the major threat was for injury. And when you're injured, you need a lot more immune cells to proliferate. So you need to produce those cytokines, the signaling proteins, that give the signal for those cells to proliferate. When that happens within the brain, it can be bad news. Now, there are several mechanisms of inducing inflammation and other mechanisms that block inflammation. And these work together in a normal condition to balance the amount of cytokines to the extent that new cells will develop, but not too many, and that the concentration of pro-inflammatory cytokines will not be too high to endanger the organism. What happens in neurodegenerative diseases is excessive formation of these proteins. Now, over the past 10 years or so, it was discovered that the main blocking system of inflammation in the mammalian system is through acetylcholine. So the same neurotransmitter that serves as a communicator between nerve cells, or between nerve cells and muscles, or nerve cells and glands, and so on, is also reacting with acetylcholine receptors on the surface of macrophages, the immune cells, or of microglia, or of nerve cells, or of muscle cells, and it blocks the production of pro-inflammatory cytokines. Now think about Alzheimer's disease, where we started our story by saying cholinergic neurons are the first ones to die. So the depletion of acetylcholine also must involve excessive inflammation. Now, inflammation markers increase with age throughout the body in all humans. This is an age-related mechanism. And you could look at that as a feedback reaction to the accumulation of insults just because we live longer. We have more opportunities to be exposed to insults. But if the inflammation markers go above a certain level, then that by itself will cause edema, will cause swelling of the brain, and will increase the risk of more nerve cells to die. So again, one needs to balance. You don't want the immune system to block the inflammation response too much. Because if you do that, then you are at risk that when the next bacterial invasion will occur, the patient will die from pneumonia. And indeed, this is the cause of death of most Alzheimer patients. Alzheimer's disease by itself does not kill. What kills the patient is another disease. So there is always this interplay between the need to block inflammation and the need to protect the immune system to confer sufficient capacity to fight invading bacteria. And what you see here are those microglia that show a lot of inflammatory markers, and the nerve cells surrounding them, and the Alzheimer's plaques that are killing those nerve cells. What are the mechanisms that protect against inflammation? These are not simple, but I'd like to show it to you as an example for those molecular cell biology studies that try today to address the mechanism so that in 10 years there will be new drugs under development. So we are talking about microglia as the immune cells. We're talking about the nerve cells as the target. We're talking about astrocytes as the communicators of signals between microglia and nerve cells. And what are the mechanisms? The first mechanism involves the production of pro-inflammatory cytokines. Interleukins, mainly interleukin-1 and interleukin-6, but also others. Tumor necrosis factors. Again, you see the overlap between cancer biology and neurodegenerative disease. These are two major fields in life sciences, and there are always focal proteins that share both. There is a new gene, NUR1, which is around which this paper is focused, so you can look for that in Cell. And this gene was shown in that particular paper to block the inflammatory reactions of microglia. Now, when that happens, the astrocyte will react to the change in the inflammatory markers, and it can communicate, thanks to that, both with the microglia. MCSF is a proliferation-associated protein that is known from leukemia. And it affects microglia because, as we said, these are actually immune cells. So the astrocyte communicates back with the microglia saying, start proliferating because there is lower levels of cytokines. So we need to rebalance. And the astrocyte, on the other hand, sends signals to the neuron. Which are those signals? Nitric oxide is one of the signaling molecules that are most studied in the nervous system. Because nitric oxide has been used, maybe it is still used, by dentists. It's called the laugh gas. Yeah, so dentists used to give it to patients because it dumps the feeling of pain, which reminds me, I need to call my dentist, but she doesn't use the laugh gas. And NUR1 also blocks nitric oxide. So the same gene can block the astrocyte response and the microglia response. It can block production of cytokines. It can block the production of nitric oxide. Nitric oxide regulates the production of reactive oxygen species, ROS, reactive oxygen species. So we block the oxidative stress together with inflammation. We'll talk soon about oxidative stress as the next complication in Alzheimer's disease. What happens to the neuron is that when it gets too high levels of nitric oxide, it would die. If it gets it from the astrocyte or when it's produced in the microglia. When that happens, the neuron will send signals of phagocytosis. So if a cell dies, you need to clean down the remnants. So phagocytosis, which again we know from the immune system, is now becoming a focus of interest in neuroscience. And these would be the more detailed mechanisms of how NUR1 sends these signals, and it does that through the transcriptional activator NF-kappa B, which again we know about from the immune system. So this is the kind of flavor of studies that are done today, and you will only see that in pharmacology in 10 years' time. Yeah?
Can I just ask you a question about this
Sure.
study? I'm interested to know whether the NUR1 gene, is it only expressed in cells that are in the central nervous system? Or
Well, this particular paper was in the central nervous system. I'm quite convinced that it is expressed elsewhere, but it's a new paper. So again, that would take 2 or 3 years until you find it in other journals, in other systems. In principle, nerve cells express at least double the variety of genes and proteins compared to other tissues. And that is are at high levels here, low levels there. And again, this doesn't say that they don't play an important role there. So never assume when you see a paper that this is limited to a particular tissue. Yeah.
This is not specific only to Alzheimer's, right? This is general mechanisms of inflammation and anti-inflammation,
True.
which can affect many diseases.
Definitely. In the link to Alzheimer, I think we did mention it last week, is that 10,000 people received anti-inflammatories to avoid heart attacks, and the outcome was a lower risk for Alzheimer. So that made this a focus. Okay. So we are talking about apparent connections between the phosphorylation of tau and the deposition of amyloid plaques, which is called in this review the different beliefs between Taoists and Baptists. So, A-beta or Tau believers. I checked, and the number of articles on Tau is about 5% of those on A-beta, which could be at least partially because this is a newer field, but maybe the believers are not that many in this particular mechanism. In both cases, this would lead to neuronal death and dysfunction in the opposite order, so first dysfunction and then neuronal death. Okay, and now we'll move into oxidative stress. So what is oxidative stress? It would be the production of reactive oxygen in excess of the antioxidant mechanisms. Again, disbalance. The newer definition would be altered homeostatic balance resulting from oxidant insult, which is almost the same. And what is oxidized? So if we look at the Alzheimer brain, we find that the answer to this question is everything. We look for lipid peroxidation, and in Alzheimer's disease there is much more lipid peroxidation, as you see here, as compared to the normal brain. We look for protein oxidation, so free carbonyl group in proteins or H&E adduct in lipids, and again we see much higher oxidation in the Alzheimer brain. We look for nucleic acids. Nucleic acids can be oxidized as well. And again, we remember that most of the nerve cells we have will not be replaced. So when they're oxidized, this is bad news. And my friend Musa Yudim from the Technion, after this slide, would tell you that's why you need to drink green tea. Because green tea is one of the strongest antioxidants known. And Yaakov Hochman from this institute said, no, green tea, nice, but what you need to do is drink cranberry juice. And they're probably both right.
But you told them that the correlation does not imply causality?
Wait, we'll get to that. Actually, there are studies, and for example, I recently reviewed a PhD thesis from that lab in the Technion, where they showed that in cultured cells, they can prevent the damage to nerve cells by avoiding the oxidative stress.
I know that in whole organisms, in all the models tested, there was no success in either preventing symptoms with antioxidants or the other way, making harder symptoms by removing SOD genes or
And
those
still people try to study that.
But I like tea, that's why.
Yeah, I mean, green tea is nice. And of course, sugars are oxidized. So the damages are there. And they occur to all of the constituents of the brain. Whether or not this is a cause or could we reverse it, would that be therapeutic? I don't know. But what is the cause of the reactive oxygen species was studied a lot. And again, redox active metals. We did talk last week about aluminum. So the aluminum story gets up all the time. And other reactive metals become popular. The last decade, people are studying the toxicity of copper-derived oxidative processes. And I know a company that is now, I think, in the third phase of curing us all from the copper damages. But anyway, the amyloid beta peptide is considered as a cause for oxidative stress. Malfunctioning mitochondria, this is important because this is the energy factory in all of our cells. Active microglia, which are hyperactive and can create reactive oxygen species, and advanced glycation end products. So again, the oxidative stress might have a feed-forward effect, because once you create it, you amplify the capacity of the existing materials to create more reactive oxygen species. Mitochondria are very interesting. There's a lot of studies relevant to aging in general and to specific neurodegenerative diseases in particular. They talk about damage to mitochondria as a major threat and cause for short life expectancy and susceptibility to disease. Again, there was a company that I'm not sure if it still exists in California, which the material they used for research was a cell line which they could deplete from all mitochondria and replace them with mitochondria from tested individuals. And the idea was to check the way your mitochondria can be cured from whatever not. But as I said, I don't think that they survived. The mitochondria DNA suffers deletions that are particular to neurodegenerative disease, and the deleted DNA is highlighted in the Alzheimer brain. And the interesting thing is that all of these damages seem to take place in the same sites within the brain. So what was done in this study was to look for the distribution of increased neuronal levels of mitochondrial DNA, the adducts of lipids that were oxidized, nitrotyrosines, which is the sign of oxidation of proteins in Alzheimer's disease, and what they found was real overlap. So cells where lipids were hyperoxidized also had, some would say, not surprisingly, mitochondrial DNA that was subject to damage and proteins that were hyperoxidized. And this shows you the mitochondrial components, and what you see here are those phagocytic vacuoles, the autophagosomes, which are the latest word in a lot of neurodegenerative diseases, that are encapsulating the damaged material to take it away. And that reminds us the discussion about the amyloid fibrils, where researchers are still arguing, is this the damage-causing entity, or are the amyloid fibrils an effort of the brain to pack together and remove the damaging material so that it won't cause more damage? And of course, mitochondrial damage, like tau, would create problems. Would cause damage to the axonal transport from the neuronal cell body to the synapses. And what you see here is how this would show the mitochondria produce energy. The energy enables transport of material from the cell body all the way to the synapse. When the mitochondria are sick, then you see a depleted transport. And the important point is that we inherit the mitochondria not through the DNA of father and mother, but only from the mother. Mitochondria are inherited only from the mother of individuals. So if you want to know the prospects for disease, you need to look into your maternal origins rather than the total genetics of the family. And we'll stop here and get together in 15 minutes. היי ביביאנה, מה שלומך? מה קרה? אני יודעת, טובי אמר שהוא ישב אתמול כל היום במקלט. אני יודעת. אני לא באה. אוקיי. ביביאנה, תלכי הביתה, אני מקווה שתוכלי לבוא מחר. 100%. כן, אני שמעתי ומבינה, בסדר? שרק יהיה טוב. אוקיי. ייגמר, אנחנו כבר היינו בסרט הזה, ייגמר. 100%. כן, אני יודעת.
ואני הבנתי שזה לא פשוט.
וואו. כן, גם המזכירה שלי איננה, כי הבת שלה גרה באשדוד, אז היא הלכה לטפל בה בנכדה. עשרות ביום, אני יודעת. מאה אחוז, ביביאנה, להתראות, תודה. הלוואי. אוקיי, ביי. תגידי, הכלבה חזרה?
שאם עדיין נכנס לגנום, למרות שהוא אמר שזה דרך אבל הוא אמר שזה דרך
ואז היא ברחה, ואני עושה את זה גם לי. אוקיי, אין דבר. תשאירי את השער פתוח שהיא תוכל להיכנס. כן. כן. אין דבר. אוקיי, תודה. אנחנו בודקים. 100%, תודה לך.
אני יכולה לדבר לך?
כן, להתראות.
average was high.
Yeah. It was interesting that further grades,
Yeah.
like a lot of people did well, and then there were like people who had like a 30 on their too.
Yeah. No, but it's not, oh yeah, well, he did publish the exam grade.
Right.
Yeah, I look at the statistics. Well, the test was a
I just think like if you just
Thank you.
No, that's really good. You have to ask them on every single question.
yeah.
I don't like to do that.
Yeah, but you needed to for this exam.
Now you
Yeah, great. I had one exam, I literally asked about every single question. So the second time I called him.
Not annoyed, he was
amazing.
he was easy.
Really
It was so clear.
wow. Yeah. So he was really okay.
Earlier today, there were a few people I think who were
Oh, I did, yeah. Why? It's the same exam.
Yeah, no, it's exactly the same. It's just that he would make announcement like every 2 minutes in our exam somebody would ask a question and then he would make an announcement about the question and about so I would assume he might have done even better if you had just made everyone else was
Oh yeah. No, it's fine. I got what I wanted. Another course that I yeah, it is. Really. Oh, where are you thinking? Really? You're not sure? No, it's if you're not. Even for courses you want.
just sign up for them at the day, it doesn't matter.
You're not paying? How come? So how do you do it? What?
I was doing prerequisites for the last year, and some research, and I applied for this done with the pre-med prerequisites.
Do you have a first degree?
I have a master's degree. I wanted to.
When you're a doctor?
Yeah.
Yeah, when you're
לנסות להיכנס לשוק שיש מחירים הרבה יותר תחרותיים לחברות אחרות. הם חייבים להתחיל ממחירים נמוכים, אחרת אין להם סיכוי בכלל לחזור, נגיד לשוק האנגלי ולשוק האמריקאי, ששם יש להם תחרות. זה לא שוק שהם מבוססים בו כבר לוקל.
זה אחרי.
כן, אני לא בטוחה.
Okay. So we spent the last hour telling that all of the mechanisms are related to each other. And here is another example. So what we do here is to immune label those stress-induced kinases that we tried to inhibit before. And what you see here is that all of the pathways that we know about as stress responding, again, mainly from cancer studies, like the ERK pathway, the JAK pathway, the P38 pathway, all of the kinases that are relevant are highly induced in the Alzheimer's brain. Okay. So from this we move into the complicated issue of animal models. When you develop a drug, by today's rules at least, you cannot start from testing a drug on human beings. You first need to show that it is showing efficacy in an animal model of the disease you'd like to treat. And the problem is that mice do not develop Alzheimer's disease. They don't have amyloid plaques. Nobody grows mice and studies the aging, mainly because it's very expensive to pay the animal facilities, I suppose. But the only animals that show amyloid plaques and signs of dementia are primates. And you don't really do drug studies on chimpanzees because it's both expensive and ethically problematic. So the solution that people try to find is to create animal models by engineering a strain of mice that would express a mutated gene. That by itself raises a very important issue. We started last week by saying 95% of the patients do not carry one specific mutation. Yet all of the animal model mice are based on creating a mutation. So are we developing drugs only for those 5% patients who have the mutation? Or are we assuming, as I think is the case, that those mutation carriers reflect the symptoms that are shared by everyone, and therefore anything that will cure a mouse with that mutation should be good also for the sporadic patients. This is the assumption, and it's by no means proven. So that we need to remember. Okay, so how can we create a mouse model of Alzheimer's disease? And what can we do when we have such a model? We take a mouse, suppose we have that mouse, dissect the brain, prepare samples, fractionate them, and then we can run gene chips. We can ask what is the transcript profile. Today we will do that by power sequencing, not by gene chips, that doesn't matter. We can use mass spectrometry to look at the composition of basic constituents like lipids or sugars. We can run two-dimensional gel electrophoresis or use more advanced mass spectrometry tools to look for differences in proteins all the way down to changes in the post-translational functional modification of protein. So not only the protein, but to what extent it is hyperphosphorylated, as is the case with tau. Or we can use microarrays with different antibodies to look for changes in the protein composition. So this would be the first test, and when we know that, we will go back to the human tissue. There are brain banks of Alzheimer's disease patients who donated their brain pre-mortem, and the brain tissue is prepared very rapidly and very professionally, and is available for the scientists worldwide to get. It's a long process, and you need to prove that this is for research purposes only, in a confirmed institution only, etc., and pay the expenses, but it's doable. And then you say, I found a difference in the mouse model, and I will look for whether that difference also takes place in the human brain, or in cell cultures of human origin, or I'll try in vitro to check whether my drug would change that modification that I found in that constituent. So this is, in essence, what one would like to do with the mouse model. The very first mouse models for Alzheimer were based, surprise, on the cholinergic hypothesis. So what people do was to dissect the cholinergic basal nuclei, which created a lot of damage, or to inject a toxin which is bound to a receptor that only recognizes cholinergic neurons, which again intoxicates those cholinergic neurons. But then genetic engineering developed, the Alzheimer-related genes were found, and people started preparing mice which carry the mutated version of the amyloid protein, the mutated versions of presenilin, the mutated versions of tau, and combination of all of these. So this is what was done. Families of patients were found with mutations in the amyloid precursor proteins which induce Alzheimer's diseases with an early onset. Very well-known examples are the Swedish mutation, which is a double mutation that occurs here between the cutting points of alpha and beta secretases. 2 amino acids are modified, and carriers of that mutation all develop Alzheimer in a very early age, and the mutation, apparently, it was formed in Sweden, and it exists in several very well-recognized Swedish families. And, of course, all carriers will develop amyloid plaques, and most of them develop Alzheimer before the age of 50. Then there are mutations that enhance the hyperphosphorylation of tau. And those, I think, were found in Italy. And then there is another family of Germans who live on the Volga. So German-originated families that immigrated about 250 years ago to the Volga region, and they live since then in Russia, but they were all inbreeding because they wanted to get married to someone who speaks their language. And that created a continuation of that mutation. And there were mice that were formed with all of these mutated genes. What was the outcome? So you could create a mouse with the amyloid protein, or a mouse with the presenilin. Remember, the presenilin gene is a component of the secretase complex that cuts off the amyloid protein into the neurotoxic peptide. Or with mutated tau, and then you could mate these mice with each other. What happens with the APP mouse is the development of plaques, at the age of, this is how it looks at the age of 4 months, 6 months, and 1.2 years, these mice brain is really full of blood. They don't show that much memory and learning deficits, but then rat investigators would tell us that's not surprising because mice are stupid animals. Rats are much more sophisticated. The problem is that genetic engineering of rats is not very well developed, and they are big and vicious animals. So people don't really like them that much. So this is the most abundant model. The APP together with the presenilin, and the tau mice, as you see, show tangles in different regions, not in the same regions where we see the plaque in the amyloid mice, but today there is a commercial line with everything, all three, and they show plaques from the age of 2 months or so. Okay. So, yeah, sure.
Do the tau mice also not show deficits in memory?
They all show deficits, but they are not as dominant as in human beings. That's the issue. Okay. What do you do to learn about learning and memory in these mice? So there are very different types of tests. You could check swimming patterns. You could put in a bath a platform where the mouse could escape the need to swim. All mice can swim, but they don't like it. So a normal mouse, after a couple of tests really, learns that over there on the right-hand corner of that bath, which he recognizes by signs on the walls, there is a navigation system in the room where the mice are tested, and they will immediately swim to where the platform should be. Even if you remove the platform, they'll swim over there. The mutant mouse would take a much longer pathway. They don't remember. They drive back and forth. I once said in a lecture that the mouse goes back and forth because it forgot its glasses, it doesn't know where the keys are, and a woman in the audience said, just like me. So careful not to say that with an aged audience. Anyway, this is how they improve learning. They really improve very, very rapidly. Okay. Then there are the different mazes. A mouse can learn to choose an arm of a maze. This is a Y maze, because they remember that over there there'll be food or something sweet to drink. And the mouse that is demented will forget that. So they will go less frequently to the correct arm of the maze. That can be made much more complicated, and all of these systems go together with video cameras and computer algorithms to calculate the extent of deficiency in the behavioral pattern. And you could check whether a mouse prefers a new element compared to an old and recognized one, so familiar object or novel object. And again, the control mice will learn that much better than the mutant mice. So there are different learning and memory tests. But how do we know whether that goes together with the pathology? And how do we diagnose Alzheimer's? That was a real problem for a long time. For a long time, you would read scientific papers saying Alzheimer's disease can only be diagnosed post-mortem, which of course is a bad thing. It's not only diagnosed late, it's diagnosed much too late. So first, there were brain imaging technologies developed. When you can see the loss of material in the brain, that's a very important hallmark for diagnostics that can be done earlier. But then scientists wanted to see the amyloid plaques as they form in the live brain. Because the belief was that this, or maybe it still is, if this is the cause of disease, then let's develop a way where we can put a patient in a machine and tell them, you have plaques to this and that extent. So they took a compound, researchers in Pittsburgh took this compound, a benzofuran derivative, and they made that into what is called the PIB probe, the Pittsburgh Initiative for Beta Amyloid. Okay? And they developed it in a radioactive version. And today it is possible to have a patient injected with the PIB probe, put them in a PET machine or in an MRI machine, and get a map of the plaques in the brain. This was not confirmed for use in Israel. The Ministry of Health says we have much more important priorities than finding out that a patient who is demented because they have plaques. In the United States, it became the prevalent method. In Israel, not yet. The machines are very expensive. But we are talking about how do you develop a new way for diagnosis of an important disease. And what was needed was an animal model proof that this probe indeed labels the amyloid plaques. So first, they took the brain of those transgenic mice and prepared homogenates and incubated them with the ligands and determined the constants of interaction. Then they prepared autoradiographs. So these are brain sections that are incubated with that probe. And finally, they put the mice in a small animal's MRI machine. We have one in Hadassah. And what they found was that here are the precipitates, and they also accumulate next to blood vessels. So that was the development, and now we have a way to follow in the animals the accumulation of amyloid plaques, and of course in human patients. So not only therapeutics development is complicated and expensive and involves a lot of elements, but also diagnostics. Now I'd like to show you an example. Yeah, please.
Didn't you mention last week that you can see plaques also in many people who don't have Alzheimer's disease?
Absolutely.
So this is not like the specificity of this as a diagnostic tool shouldn't be very high.
Well, 40% of the people who show plaques do not show dementia, and the argument is it because this is too early, or because they have ways to avoid the damage, or is it because the amyloid plaque is a protection mechanism and not a damaging one? This is still not known, but the method is used. Okay? So next I'd like to show you an example from our own study for a variety, and because I know it better than other methods. So what we've done in the 90s is to clone the human cholinesterase genes. And the idea was, if cholinergic neurons are dying, then cholinesterases could be either damaging agents, which is why we need cholinesterase blockers. These are the leading drugs today. Or maybe they can protect and they are lost because the cholinergic cells die and they also produce cholinesterases. So we cloned the genes and we created transgenic mice that overexpress one or the other. And at the time I wrote a patent saying we have an animal model that produces cholinesterases in its milk. We really milked mice, imagine. And we determined the activity in the milk, and the enzyme was expressed in all tissues, so also in the milk, and they confirmed the rights on a mouse producing cholinesterase of human origin in the milk. Now, nobody would use a mouse to produce a protein. The idea was this is a production system. So we tried to argue, and we were not very successful. But luckily, one of my ex-PhD students owns a company for writing patents in biotechnology. So we approached him after we were refused once, and he prepared a folder like so, showing that a mammal is a mammal is a mammal. If a mouse produces it, then a larger mammal would produce it too. And finally, we got the patent. And then a company was established in Annapolis in the United States. The registered value of that company is they own a herd of engineered goats that produce human butyryl cholinesterase. This is one of the cholinesterases in their milk. So they went to Africa, they found a kind of goat that matures very early, so it's like at the age of 8 months, they already produce the next generation, and they made them transgenic, and they purified the protein from the milk. And the next thing was I was going for sabbatical, so I had to get a new visa, and I went to the consulate in East Jerusalem and waited a whole day, and finally I get to see the consul, and the lady consul says, okay, you want a visa, this was a fellowship visa, which is complicated. Tell me the reason for your most recent visit to the United States. And I said, well, there is a company in Annapolis, and they own a herd of engineered goats that produce a protein for which I clone the gene, and the university owns the rights. And she looks at me and she says, what? And I said, you want a letter to confirm that I work at a university, which I did not produce before because I was the dean then. So in our university, such letters are signed by the dean. And I also had a letter where I signed that I work where I'm staying. So this was really complicated. And she looked at me disgustedly and said, no need, lady. Nobody can make up such a story. So this is the study I want to tell you about. These are the goats, okay? The company is called Pharmacine, and they are in Annapolis. It's a very nice place. And our study was, I was very pleased that they produced this protein. They produce it as a weapon or as a defense mechanism against poisonous nerve gas, which is targeted against cholinesterases. But I wanted to use it for an Alzheimer purpose. So what we did was to ask the question, what happens when this protein, the cholinesterase, which is missing in the brain, interacts with the amyloid peptide? So if you take the amyloid peptide by itself and incubate it in a solution and then go with electron microscopy and check what happens, the peptide immediately creates fibrils. It's a very rapid mechanism. So this is the electron microscopy map. If you incubate it with the protein, which is produced on chromosome 3, also on chromosome 3, then the production of fibrils is arrested. So this protein serves as a protection. And actually, its levels go up in the Alzheimer brain, probably as an effort of the brain to protect itself. Only that the American Army didn't continue their payments because when you inject people with this protein, which they did, they develop antibodies to the goat glycosylation system. So right now, the company is there, but they don't have any more funding. So this is an example of how you try to look whether a protein that increases in the brain is protective or damaging. And this is the student whose PhD was on that, and he's now with Roger Kornberg at Stanford doing postdoc. Okay. And then we asked, why is that? How can this protein protect the amyloid peptide, preventing the formation of fibrils? And would specific variants of this protein behave differently. So 20% of the humans we know carry a variant of butyrylcholinesterase with one single mutation very near the C-terminus, which doesn't change the active site capacity. So that is a protein that can hydrolyze acetylcholine just as well. But the C-terminus has 1 point mutation in it. Carriers of that mutation develop Alzheimer later than others, but when they do, they don't react to therapy. And the reason for that was not known. So the mutation is on tyrosine 8 in the tryptophan 8 in this peptide. And this is called the K variant because it was discovered by a guy named Callow. So this is the guy who discovered the mutation on chromosome 3. Over here, there is a switch of one nucleotide from A to G, and that changes the amino acid. Okay. So now we look at the activity of cholinesterase in the serum of carriers. These are the normal carriers, and what you see here is a distribution of activity. So what percentage of tested individuals would have this activity? So what you see here is a Gaussian distribution. Almost everyone has that activity, and some would have less, and some would have more. If you look at carriers of one copy of the gene with the mutation, you don't see much of a difference, but homozygotes have a real difference in activity. So how come they have a lower activity when we know that the mutated protein can hydrolyze the substrate just as well? The mutation is very far away from the active site. Anyone has a suggestion? A protein with the active site here, a mutation there, and somehow when we look at it in real people, we see that the activity is lower, whereas if we compare the protein in a synthetic way, there is no effect on activity. What went wrong?
If you look at the transcription level, is it the same? Because
The same transcription.
Something can bind to the mutated site.
Bravo. The interactions, the protein-protein interactions are modified. So, okay, so these are the activities, and this is the peptide, and what you see here is the mutation, and what happens is it changes the interaction of the folded region at the C-terminus. And what we said is we have to look at what are the reasons for the change in the properties of this protein, and we did that in two ways. First, we incubated normal or mutated protein, which we have from the serum of these carriers, with a synthetic amyloid peptide. And we check how long does it take the peptide to create a fibril in the test tube. And we check that by running gels, and what you see here is that when you run gels, this is a monomer, it's labeled with antibodies, and this is a dimer and tetramer and so on. So you have formation of multimeric versions of this protein, and then it disappears. And when it disappears, it appears in the pellet, so it precipitates. Okay? The peptides interact with each other to create pellets. And what you can see here is that the disappearance is much faster, these are time in hours, for the K carriers, okay, as compared to the normal people who have the U variant. So something behaves differently. This protein interacts with itself or with the amyloid peptide or with both differently. Why? So to check that, we used nuclear magnetic resonance. When you have a nuclear magnetic resonance machine here in the Structural Biology Center. You can put a peptide, and you can synthesize the peptide with or without a mutation in the machine and look at the signature of the nuclear magnetic resonance pattern of that peptide. And what we found was a difference in the folding pattern of those peptides, so that the peptide is proline rich. Proline is an amino acid that creates folding of peptides, and that facilitates the formation of tetramers of the monomeric subunits of this protein. This is under normal conditions. In the blood, it interacts with other proline-rich proteins which can stabilize or not the butyrylcholinesterase. And the mutation impairs that interaction, and therefore the mutated protein is less stable. So when you see lower activity, it doesn't mean there is a smaller amount of protein, but it rather means that that protein is there, but it's not active anymore because it creates abnormal structure with other proteins. So here is the signature, the fingerprint of this peptide, and we also looked at the structure, and here is a model of the structure. So the protein itself creates tetramers. There are 4 folded C-termini of the 4 subunits, and this is a proline-rich peptide which exists in the blood and interacts with that protein. No protein ever lives by itself. Proteins keep interacting with each other, with nucleic acids, with lipids. But the abnormal protein creates a kink, you see here? It does not create just a loop, a helix, like the normal protein, but it creates a kinked helix, which impairs its capacity to interact with other proteins in the blood. And then Eretz created this cover figure, which the journal unfortunately did not choose, which said what determines the capacity to create the amyloid fibrils is the cards you inherited from your parents. If you carry that mutant, then the formation of the fibrils will not be that good. That will delay the onset of symptoms, but when the symptoms arrive, the reaction to drugs will not be very good. Okay, so here is an example to the complexity of what we see and what it implies. And I'm not sure that this is the end or this is the only reason why that variant is impairing the reaction to drugs, but it's an example. Okay, so now we go back and we ask, is the mutation the only reason? And I'd like to argue, and that's another project of another student in the lab, Amit Berton. He's now in Philadelphia. And what he did was to go back to alternative splicing. Now, the Mac made some difference. Okay. What Amit focused on initially was the capacity of the acetylcholinesterase gene to produce more than one protein through a process called alternative splicing, okay? Which again changes only the C-terminus. In this case, it changes 24 amino acids out of about 600. And the normal protein binds the synapse, sits there, and degrades acetylcholine whenever it's there. The variant protein has a different C-terminus. It cannot bind anything. And it's a monomer, unlike the tetrameric versions that you saw. That protein, by the way, is produced in a totally different system. It's produced in a company called Protalix in Carmiel in plant cell cultures. So right now we are much more interested in this particular variant. Anyway, what Amit did was to ask how would the two variants interact with amyloid peptides. And what he used for that, instead of electron microscopy, is a fluorescent label, thioflavin. And what you see, I'm afraid you see it in a very small scale, but I hope you can. This would be the interaction, or when you see an increase in that figure, that means we get a lot of fibril formation from the amyloid peptide. And this is an amyloid peptide to which we added the synaptic version, the normal acetylcholinesterase. Now we add A beta alone, and you see that the synaptic acetylcholinesterase facilitates fibril formation. Okay, because the rate of accumulation of fluorescence is slower when we don't have it. But if we add the recombinant protein that is monomeric, then we slow down the fibril formation, quite like what happens with pyruvate. So A beta interacts very well with synaptic esterase, and that agrees with papers of others from different systems, and the disease-induced esterase does not interact with it. Okay, so there we said fine. Suppose there is a process of alternative splicing whereby the brain changes one protein to another. Would that change the development of disease? So we could take the amyloid mouse, the mouse carrying the Swedish mutation, which develops plaques, which we just discussed, and look at the plaques. This is a section of the mouse brain, and you see the plaques. These are like the ink dots spread all over the brain. This is a sick mouse. It's 8 months old, and it developed a lot of plaques, and it already shows learning and memory impairments. Now let's mate this mouse with our mouse that produces the monomeric esterase that slows down amyloid formation. And the question would be, would that change plaque formation in an animal model? And this is how the progeny looks. We do have much fewer plaques that are formed. So would that mean that alternative splicing is part of the disease? Probably. Would that mean that removing the plaques is a protective mechanism? Probably. So based on this study, we started thinking two things. First, that the formation of plaques is really a bad thing, not a protection. And second, that alternative splicing may be involved in the disease mechanism. So here are the plaques in the brain. And the idea then would be, by the way, this is the clock test. If you ask an Alzheimer patient to draw a clock, the deterioration in the capacity to do that function is considered a hallmark of the disease. So what we discussed was polymorphisms, right? Change of one nucleotide in a gene can change the reaction to the disease therapeutic, alternative splicing, and alternate promoters we didn't discuss, but it was mentioned, of course, this is highly involved as well. And that brings us to the philosophical issue of what is a neurodegenerative disease. And one might say that our nervous system is highly complex, a lot of things that should work together. With aging, this bit goes bad, that bit goes bad, and when too many pieces go bad, the system collapses, and you'll have a neurodegenerative disease. But there's another way to interpret it. And the other ways would say, true, the system is highly complicated. True, with aging, there are damages that take place. And whenever a damage takes place, we activate a correction system. And it is only when the correction systems fail that neurodegenerative disease will happen. And what I would like to argue is that here is a protein which acts to protect, and it's produced through alternative splicing. So maybe that is one correction system. Here is a protein that increases in the level in the Alzheimer brain. So maybe that's another protection. So in other ways, we have an intimate involvement of the cholinergic system constituents in the disease process and in the protection from it. So how can we check that? Yeah.
Actually, you showed the mice, the previous slide. Did you see any behavioral change in the next generation mice? Like you cross between two things, right? So the next project, did you compare the behavioral change? Because you told the behavior.
You're right, we should have, except we did that with collaborators from Switzerland, and they stopped growing the mice when the student finished their PhD. I'm sorry, you're right. We regretted that many times. So we don't know the final answer, but that caused us to go back and ask the question, to what extent would RNA metabolism impairment be part of neurodegenerative disease? So we can look in the literature, and we find there that 95% of the multi-exon genes would go for alternative splicing. This is the explanation for the fact that we have about 20,000 genes but 250,000 proteins, because there is so much alternative splicing that changes the composition of the protein products. Now, a major part of the disease-causing mutations that we know about today affect the splicing-related proteins. And I should add here that there's at least 150 genes and proteins that are involved in the splicing process. So it's a very demanding process. And mutations that affect RNA processing are primarily causing neuronal disorders for all of the reasons that we discussed. Because we have that many nerve cells, because we'll die with those nerve cells, they don't get replaced very easily, they're sensitive. So splicing is prevalent, damaging splicing causes disease, and those diseases would mainly affect the nervous system. So how can we check that? And what Amit did next was to use a multi-exon array. So you take a glass slide and you put on it dots, each of which carries a probe for one exon, not one gene, but one exon. Altogether, 1 million exons, and for each exon you have several probes, so 5 million probes on a chip. And then you can check all of the genes in normal and Alzheimer brain and ask, is alternative splicing changed not in those mutation carriers, but in those sporadic patients that developed Alzheimer for unknown reasons? So we remember that Alzheimer's disease was cholinergic to begin with, that the amyloid hypothesis commenced, that we have tau and neuroinflammation involved. Now we are asking, are RNA metabolism errors involved? And this is the comparison we did. Yes?
I may play the devil's advocate.
Please.
You're looking at brains of two different individuals, one with the disease
Not
and one
two.
without.
Not two,
Okay.
several of each.
Several of each. But still, the variance between humans is considerable. And you're looking at
No
millions
doubt.
of probes, and you need to correct for
human beings are not inbred animals, unlike our transgenic mice. You would say, suppose I use this technique and I take 20 of this and 20 of that, and I see a significant difference in a process, then it should be right.
I noticed in MS, for example, which we
Okay.
talked about earlier, so several groups around the world did it, and everyone did it not on dozens but on hundreds of patients,
Okay.
not this generations of chips, but the one before that, and everyone got hundreds of significant genes, and the overlap between them was 0.
Of course. But again, that agrees very well with what we assume, because we have 20,000 genes, but that many functions. So you could use changes in different genes to bring about a modification in a function. What creates the disease is depletion of a function. Okay? And that's what we ask, whether it happens or not. So what Amit did was to ask, are there genes that are overproduced in the Alzheimer brain? And he found genes that agreed with all of those previous publications. Are there genes that are underproduced? Again. But then he also found a third population of genes that were not up or down regulated, only alternatively spliced. Okay? And they were not alternatively spliced at random, but what they showed was an added exon. They showed exon inclusion, so a function was changed. I'm not talking about a gene now, I'm talking about a pattern. And I don't believe chips. That's another problem. So we did a lot of validation. Here you see 2 genes, for example. These are the structures. And you see there are many, many probes for each gene. And in one site, we see a difference between Alzheimer and control. And here's another one, same thing. And here are the products. You see that in the Alzheimer brain, you always have a larger product. And that happens in about 300 genes and 383 exons. So in most cases, there's just one exon that is modified.
Which brings again to the problem of causality.
Again, absolutely. Correct. So we did the validation. We said, okay, this seems to be a phenomenon. There is a massive exon inclusion in the Alzheimer brain. So who's responsible for exon inclusion? There are 2 major gene families. There are the SR proteins that control the inclusion and the HNRNPs which control the inclusion. And this is inverse to what happens under stress. Under stress, nerve cells react by expediting exon exclusion. And what we said, wow, maybe Alzheimer is the failure to react to stressful insults. If that's the case, then we need to do the experiments. And we need to look at that at the level of the protein. So we ran a lot of protein blocks. And what you see here is that indeed the proteins in charge of exclusion are gone. They are really gone from one part of the cortex, not the entire cortex, but the entorhinal cortex, which is the region that is damaged first. And then I said, okay, we don't believe one method. We need to look using another method. So we immunolabeled sections from the brain. And what you see is that the SR proteins are there, but the HNRNPs are gone. And they all label the nuclei, so something is wrong with the nuclei that express HNRNPs. HNRNPs stand for heteronuclear ribonucleoprotein complexes. You don't have to remember that. But these are the proteins that are in charge of clipping out pieces out of the transcript and creating the final protein. So we asked, is that happening also in those nerve cells? which express the amyloid fibrils. Amyloid fibrils can be labeled, as you see here, with green. And what you see is that the SR proteins are there in the healthy brain and in the Alzheimer brain, even in those nerve cells with the amyloid protein. But the HNRMPs, the pink ones, are gone. So, we lose a family of proteins. What does that mean? Here is an example. A nerve cell traditionally includes a lot of HNRMPs. It takes care of a lot of splicing. In the Alzheimer brain, it's all blue, we lose it. People ask me often, are biologists inventors or discoverers? Which is an interesting issue. And I believe that chemists are more of inventors, and biologists are discoverers. So this golden bell is mentioned in the Bible. It was tied to the clothing of the supreme priest, and it was found like 2 months ago next to the Wailing Wall. So it was not invented, it was discovered. So we found a phenomenon that was there for many, many years, and I know the lesson is over. So I promise that in the next lesson I give, I will tell you how we check that this is or can be a causal element in the disease, and what were the tools. Thank you very much. Yes, we have a faculty member who looks at the role of this protein in cancer cells.
I mean, it's like let us say, if you compare to cancer, it's exactly opposing what's happening, right?
It is opposite.
All the mechanisms, whatever you see.
Very good question. You're right.
It works the same way, but it's opposite. So like, if you have some cancer, it's overexpressed. If you have like opposing effects.
We have a new faculty member in the Faculty of Medicine that and it works. So do you enjoy the course?
Yeah.
You learn new things.
Yeah.
You know the basics. Okay.
No. I'm new to this whole thing. I haven't read anything of this.
Yeah, but you know biology.
Yeah.
I have others who don't know any biology.
But this one is actually interesting.
Very good. Okay.
אז נראה לי שאני אגיע שם משהו מפתיח ואז אני אעזור לך. איזה חינוך, נתתי לי אמר להם. בעצם. נו, מה? אם נתחת להיות שבעצם. כן, ממש. דני מגיע ב-12, אני כבר ב-9, לפני 9.
ב-10